Hi everyone, how are you? My name is Saman and I'm studying in Teaching International University in Biology department. Today we will explain the topic from 12th grade book. Before I go through my topic, I want to review the last lesson. In the last lesson, we studied the digestive system. The digestive system, as you know, began in the mouth. We have chemical digestion and by saliva and mechanical digestion by teeth. After the food go to through esophagus. Esophagus transporting the food and connecting the mouth to the stomach. In the stomach, by digestive juice and by the muscle in the stomach, the digestion happen. And now, in our topic, we study the discovery and structure of the DNA. We study discovery and structure of the DNA. First of all, in our objective, we study this transformation experiment, DNA as genetic material, chemical compounds of the DNA nucleotide, and the double helix structure. We study nucleotide, we study the DNA, the Watson and Crick model. We mean by this the DNA model, complementary base pairing. Now, let's discuss the Grieve transformation experiment. Grieve, we have in the Grieve experiment, we use the bacteria names pneumonia. We have two types of this bacteria. We have the smooth pneumonia and we have the rough pneumonia. We call the smooth pneumonia S strain. Why we call smooth? Because around the bacteria we have capsule. That capsule it is virulence, and we have rough pneumonia. They are rough in the age because they don't have capsule. And now the rough strain, it is not virulence, and S strain, it is virulent. Let us discuss the Grieve experiment. First of all, we use the R strain. We inject the R strain to the mouse, and the mouse lives. In the second, we inject the S strain to the mouse dies because the S strain it is virulence. In the third attempt, we kill the bacteria by heat, and after that we inject it to the mouse, and the mouse didn't die. The mouse alive. In the last attempt, the last experiment, we killed the S strain by heat and mixed it with the R strain, leaving R strain. After that, we injected to the mouse. The mouse died. The Grieve said something transferred from the die S strain to the R strain, leaving R strain, and make the virulence and the mouse die. After that, we have two scientists, Hirsch and Martin. Hirsch and Martin say we have two things. We have protein and we have DNA. They try to know DNA is transferred from the dying bacteria or protein transferred from dying bacteria to living bacteria and make the bacteria virulence. Now, as you know, if you see this picture, look at this picture, you have radioactive P32 labeled DNA. We label DNA by P32 and we label the protein by S35. And we infect this to the bacteria. This is microphage and infect bacteria, and we blender the bacteria. After that, we centrifuge it. In the end result, if the taste inside the bacteria we have S35, it is shown that the protein is genetic material. But if we found the P35, we know the DNA is genetic material. After this experiment, they discovered that we have the P35 inside the bacteria after centrifuging, because that's the reason that DNA is genetic material. Now it is DNA is genetic material. Now we talk about nucleotides. You know we have a cell, we have this cell. Inside the cell we have nucleus. Inside this nucleus we have DNA. DNA it is like twisting ladder, like we make it like ladder. DNA like that. It's like a ladder. And we take a half of the DNA. We take this part. This part, it is, we say the nucleotide. In this part, what we have? In this part, and we make it smaller. We take this one. We take this part of the ladder. What we have in this part of the ladder? We have first of all phosphate group, sugar, and nitrogen base. We have these three parts. This part we call nucleotide. Nucleotide, we have first one. We have phosphate group, sugar. We have the 5 carbon sugar. We call the deoxysugar. It is different from the RNA. What is the difference between RNA and DNA? The sugar is different. Nitrogen base, we have adenine, guanine, thymine, and cytosine. And in the nitrogen base, except of the thymine, we have the uracil. This is the two difference between the DNA and RNA. First different, instead of the deoxysugar, we have the 6 carbon sugar. Except of the thymine, we have uracil. It is so important for the exam every time they take a question from this. And the Watson and Crick model. We have the Watson and Crick model here. In the 1953, Watson and Crick discovered the structure of the DNA. It is like twisting ladder. It is twisted. And if you see, we have backbone. In the backbone, we have phosphate group and sugar. And in the middle, we have the nitrogen base. This is the structure of the DNA. Now we talk about the base pairing. As we know, every time the cytosine by 3 hydrogen bond, this hydrogen bond, hydrogen bonds, cytosine join with the guanine by 3 hydrogen bond, but adenine with the thymine join with the 2 nitrogen bond. As I mentioned that if you have RNA, if RNA in the nitrogen base, except thymine, you have uracil. This is so important for the ministry question. Every time you have the question about this, except the thymine, what you have in the RNA, you have uracil. And this part, it is phosphate group. And this one part is sugar, right? Sugar. And this part phosphate group and sugar. We call this part what? Backbone. It because of backbone. In the exam, if you say to you, what is the backbone? Backbone composed of what? Composed of the phosphate group and sugar. Now let us review what we did study. First, for Greaves transformer experiment, we know that Greaves inject the mouse by S strain that killed the mouse, and R strain didn't kill the mouse, and heated S strain didn't kill the mouse. Well, mixed dyed S strain and leaving R strain killed the mouse. DNA and genetic material. That experiment that we do it in the macrophage, we discovered that the DNA is genetic material because the DNA transferred. Chemical compound and DNA nucleotide and the double helix. We know that the DNA it is double helices and it is inside the nucleus, and DNA chemical of the DNA composed of the nucleotide. In the nucleotide we have phosphate group, 5 sugar, and we have also nitrogenic base. The Watson and Crick model, this is the double, this was the Watson and Crick model. We have the twisted ladder, we have the backbone composed of the phosphate group, sugar, and bases. And after that, what we have in the last complementary base pairing every time in the DNA. Let's make like that. D and A. Adenine by hydrogen bond, double hydrogen bond with the thymine, join with the thymine. What we have? After that, guanine, triple bond, cytosine. But in RNA, adenine join to uracil by double bond, and by triple bond, guanine join to cytosine. I prepared some ministry question for you, and let us do the ministry question. In brief experiment, what specific combination of the bacteria caused the death of the mice? What caused the death of the mice? First of all, in the A, living strain. Living strain is no virulence. If you inject it with the mouse, they didn't kill mouse. This is wrong. You want a mouse to die. Let's kill the mouse. In the B, multiple choice, heating killed S strain. If you kill the S strain by heat, they didn't kill mouse because the mouse leaf. Here, mouse leaf. In the third multiple choice, heating killed S strain mixed with the living kill mouse. Yeah, kill. Because that's the reason, the correct answer here it is answer C. In the D, poor soil extract, it didn't relate to our topic because we remove it. And in this question, in this ministry question, Erish and Chase used radioactive phosphorus PCU to label which part of the bacteriophage. We used the P32 to label the DNA. It's important here. We label protein, carbon, amino acid, they didn't relate to our. And we have last one. What is the name of the 5 carbon sugar found in the every DNA nucleus? We call it is deoxyribonucleic acid. Deoxyribo. It is the name of the sugar. The correct answer is B. Now we are finishing our lesson, and for the next lesson I send you like 15 minutes equation. You have to solve it. It is like a homework for you. Thank you very much.